This is studying the pH of strong acids, weak acids, and salts from the General Chemistry Lab Manual, omitting the buffer solutions section. Okay, as you may know, acids dissociate in water, and it sets up an equilibrium where the acid loses a proton to water. I'm writing it with water present. If you wanted to write H+, that's fine as well. And then you get the anion, A-. Now the equilibrium constant then is a special equilibrium constant that we give a special letter to. We call it the Ka because it's special for acids. And it has the form where the products, as we would expect, the H3O+ and the A-, the conjugate base there, are in the numerator, and then HA is in the denominator. Water is not present because water is a pure liquid. Pure liquids have equilibrium or contributed as one, so they sort of fall out of the equation. And so that is the Ka relationship. Now, the other more common way that we know how to measure acidity is with pH, and so pH is the formula, as we've had before, is minus the log of the hydronium ion concentration, or H+ concentration, however you want to call it. And so you can break down acids into sort of two groups. So the strong acids, where it's 100% dissociated. We just assume that Ka is basically infinite because there is no HA left. And there are 6 of them, the halogens, HCl, HBr, and HI, and then as well as nitric acid, the first hydrogen of sulfuric acid, and perchloric acid. Those are all strong acids, and basically the acid concentration is the H3O+ concentration. Everything else are weak acids, where it's less than 100% dissociated, and it depends on the Ka. Some are still relatively acidic, and others are not. And then that takes into account salts. Now salts, after dissolving, potentially the ions could be acids or bases. They could act as conjugate bases, and so we'll look at some of those solutions as well. So these are the main relationships. You probably had them in lecture, but if not, there's also going to be a post-lab that'll talk about how to calculate these various things given some of the other components. So the lab is pretty straightforward. What you're going to do is you're going to make a series of dilutions to the point where we call it a serial dilution, where you'll take a solution, dilute it, take that solution, dilute it some more, and so you get this stepwise dilution of both a strong acid, hydrochloric acid, and acetic acid, a weak acid. And then you're going to measure the pH of each solution, and we should be able to predict then how that relationship would work. And then you're also going to take pH of different salt solutions and use that to deduce things about the Ka of the compound, what the species are that could potentially be acting as acids or bases. So it's a pretty straightforward lab. In terms of our objectives with general chemistry, lab objectives, you're going to learn how to do glassware, you're going to learn how to use a pH meter, but this one's really one where you're going to spend a lot of time on solutions, doing either dilutions or concentration calculations, and you'll also be doing reactions with acids and bases, so that's sort of the two major ones. But again, it's a quantitative lab, and you're going to have to be careful, especially when doing these dilutions, to get really accurate data. Now, the one thing we're going to change from the lab manuals, we haven't really had buffers. That's something that gets taught in a later course, and so we're going to skip this section on buffers. So the whole last section that talks about buffers, we're basically going to stop at the salt solutions and not do anything else. The other is that we're going to try to do these in large test tubes, but our probes that we use are not long enough to fit into 5 mils. So we're going to probably put 10 to 15 mils into each tube. The volume really isn't that important. The only time when volume is really important is when you're making solutions, especially when you're doing the dilutions. Also, they have a neutralization step where they talk about having an indicator telling you when it's okay to put down the sink. The concentrations that we're doing and the amount, as long as we use a lot of water, we can just ignore that. I do think it's a good idea to sort of pool all of your stuff into one beaker maybe and then pour it down the sink, but for the most part you can ignore that. We have enough pH meters. You can work in pairs. It's not really a problem. And from a safety standpoint, the real key issue is that HCl, especially, is corrosive. All these acids are slightly corrosive, especially at the higher concentration, so you want to wear gloves and be careful. So the key here is properly doing it. And so they kind of discuss how you're going to rinse out the graduated cylinder when you use it. Eventually, you're going to make these dilutions. You'll pour them into the large test tubes, and then you'll make a new series of it. You want to be careful with the drops. So you notice I've got a drop there at the top of my graduated cylinder, but you'll also see that I am able to sort of push that down. So be careful when you're diluting things. Make sure they are properly diluted and you're not leaving drips and everything. And then you'll do the dilutions in larger graduated cylinder. And we'll do that with DI water, and the easiest way to do it is to use a beaker and a dropper probably to get something accurate. And it doesn't exactly have to be exactly the right number. The key, though, and here I'm rinsing out the graduated cylinder too, is that you know the volume so you can calculate the concentrations. Now, the pH meter needs a little rinsing. So you're going to take some DI water and use a dropper and just kind of squirt the end of it off. Usually what we do for accuracy then is sort of blot, don't wipe, sort of blot the probe dry. We'll show you how to use the pH meters once you see them, but basically you'll turn them on. They'll already be calibrated, and we will just then stick it into the test tube. They should fit pretty well. If they don't, we might need to put it into like a 50 ml beaker or something like that, and then you can take your... So you have to remember each time to wash that probe off between runs, because especially if there's a little bit stronger acid on there, I would start at the weaker, the more dilute acids, and work my way up to the stronger acids for more accuracy. So at the end of the lab, you should have the following. You'll have your series of HCl dilutions, series of acetic acid dilutions. You'll know those concentration, or at least you'll know the volumes that you used when you made up those solutions, and then you'll get the pH. Okay, so you have that of each of those. You'll also have pH of different salt solutions. So that'll be the data that you have to have. Now, there'll be a post-lab where we can talk about sort of how to calculate Ka, for instance, from these things, from these readings. Now, the other is you need to rinse the pH probe, and I think they have little bottles you'll be putting them back into. It'll be pretty clear how to do that. And then again, we can put the waste down the sink with a lot of water. And so other than that, it's a pretty straightforward lab. Again, there'll be a post-lab depending on your instructor. They may do it in lecture, or they may choose to do it with a video. It's going to depend on your instructor, but that'll also be in place if it's needed.